Could the key to reducing brain inflammation lie within our 24-hour body clock? Recent research into Rev-erb alpha, a protein tied to our circadian rhythms, suggests this might be the case. The enlightening study uncovers intriguing connections between this unique protein, brain health, and inflammation, possibly paving the way for new protective strategies against neurodegenerative diseases. Experiencing inflammation in the brain is not merely an internal fever. It's a serious event that can trigger a cascade of damaging effects, leading to disorders like Alzheimer's and Parkinson's. Finding ways to combat this inflammation is a top priority for neuroscientists around the globe. Imagine the unfolding surprise as researchers discovered, not in a complex new pharmaceutical or diet, but within our own internal clock, a potential resource for reducing inflammation. The essential cog in this mechanism is the protein Rev-erb alpha, a critical component of our body's circadian rhythm. Making matters more interesting, this rhythm does more than dictate when we sleep or eat. It also has a profound effect on our brain's health and integrity. Engage with this article as it will unravel the exciting research insights into Rev-erb alpha and its potential neuroprotective and anti-inflammatory effects. The impact of deleting Rev-erb alpha. What happens when we remove Rev-erb alpha, a significant protein player in our biological orchestra? In this section, we aim to uncover the striking effects its deletion can bring, diving into how its absence can trigger and accelerate neuroinflammation. Rev-erb alpha deletion effects. Rev-erb alpha deletion induces spontaneous hippocampal microgliosis. Researchers observed that in wild-type mice, microglial volume varied by time of day, with increased volume observed at the time of trough expression for Rev-erb alpha-regulated targets. This effect was not seen in Rev-erb alpha knockout mice, suggesting that Rev-erb alpha may regulate genes controlling microglial activation. The knockout mice showed increased spontaneous microglial staining and increased expression of pro-inflammatory transcripts, indicating a pro-inflammatory response. They also exhibited increased phagocytic activation and decreased microglial branching, both signs of microglial activation. The total number of microglia also increased in the knockout mice, a common feature in many neuroinflammatory conditions. These findings suggest that Rev-erb alpha plays a role in regulating microglial activation. Enhanced NF-κB signaling is associated with immune activation in RKO microglia. The study further explores the neuroinflammatory characteristics of Rev-erb alpha knockout RKO mice through transcriptomic analysis of hippocampal tissue. The analysis revealed significant increases in pro-inflammatory chemokines, astrocyte activation markers, and microglial activation transcripts, emphasizing the neuroinflammatory phenotype. Pathway analysis showed upregulation of processes related to innate immune activation and inflammation. Several genes that have a role in the positive tweaking of NF-κB signaling were found to be amplified, while genes that inhibit this signaling were lowered. In RKO microglia, higher nuclear translocation of p65 was observed initially, pointing towards enhanced NF-κB signaling. The research also observed a rise in basal cytokine production in primary RKO microglia. Investigations with the help of chromatin immunoprecipitation tests suggested that Rev-erb alpha directly regulates the expression of various genes related to NF-κB. This implies that the removal of Rev-erb alpha boosts basal NF-κB signaling in microglia, pushing for a pro-inflammatory activation of microglia. Rev-erb alpha deletion induces cell non-autonomous astrocyte activation. The study explores the effect of the deletion of Rev-erb alpha on the activation of astrocytes in the brain. Increased GFAP, a marker of astrocyte activation, was noticed in the hippocampus and throughout the cortex of Rev-erb alpha knockout mice, indicating astrocyte activation. The extent of this increase, termed astrogliosis, differed among the mice, but was uniformly observed starting in the outer layers of the cortex and hippocampus from the 3-month mark. A substantial rise in the activation transcripts of pan-reactive astrocytes was detected in the hippocampus of Rev-erb alpha knockout mice. Interestingly, no increase in GFAP expression levels was seen in wild-type astrocytes after NR1D1 knockdown through siRNA or in primary astrocyte cultures from these knockout mice. Nevertheless, in response to LPS, astrocyte cultures from Rev-erb alpha knockout mice did display increased expression of IL-6 and IL-1B, indicating that Rev-erb alpha may regulate NF-kappa B in various glial cell types. From this data, it suggested that the removal of Rev-erb alpha indirectly triggers astrocyte activation, possibly through microglial activation, neuronal injury, or other factors. Rev-erb alpha deletion exacerbates lipopolysaccharide-induced neuroinflammation in the hippocampus. This study delves deeper into the neuroinflammatory reaction of Rev-erb alpha knockout mice, often abbreviated as RKO, to LPS, short for lipopolysaccharides. Interestingly, injecting these knockout mice with LPS intracerebroventricularly led to high fatality rates, which prompted a shift in the administration route to intraperitoneal injections. Post-LPS administration, significant increases were observed in inflammatory transcripts within the hippocampus of normally found wild-type mice, specifically 6 hours after the injection. The absence of Rev-erb alpha then appeared to augment this LPS-induced expression of pro-inflammatory agents in the hippocampus. This included IL-6, CCL2, the astrocyte activation marker GFAPE, and HMOX1, a transcript responsive to oxidative stress. These observations suggest that eliminating Rev-erb alpha amplifies the neuroinflammation induced by LPS in living organisms. The research sought to comprehend whether eradicating Rev-erb alpha influences the functioning of the brain. It did so by examining the cortical resting state functional connectivity using a technique known as optical intrinsic signal imaging, often abbreviated as OISI. This approach facilitates tracking alterations in connectivity across different regions within the brain. In the context of Rev-erb alpha knockout mice, the research detected a clear decrease in functional connectivity within a part of the brain known as the retrosplenial cortex. This area tends to be impacted by neurodegenerative diseases. On the other hand, an increase was observed regarding functional connectivity within the somatosensory region. Changes noted were corroborated by a quantitative evaluation using spatial principal component analysis, also known as PCA. The results suggest that the elimination of Rev-erb alpha paves the way for changes in brain functional connectivity, akin to what is typically seen in models of neurodegenerative disease. Pharmacologic activation of Rev-erb alpha. Researchers further investigated if neuroinflammation spurred by lipopolysaccharides, LPS, could be attenuated by orchestrating the activation of Rev-erb alpha. Rev-erb alpha is a distinctive orphan nuclear receptor that can be activated by small molecules like SR9009, which are capable of permeating the brain. Discoveries from in vitro experiments. The investigation delved into the possible effects of Rev-erb alpha activation on cells exposed to LPS, both in the absence and presence of SR9009, over an 8-hour period. This process was intended to activate NF-kappa B, and IL-1 beta protein secretion was encouraged through the addition of ATP in the last 30 minutes. The utilization of SR9009 revealed a dose-dependent suppression of LPS plus ATP-induced IL-1 beta protein secretion in BV2 immortalized microglial cells. Investigating the role of SR9009 with primary microglia. In the situation of primary wild-type or WT microglia, there was a clear observation of SR9009 inhibiting IL-1 beta secretion triggered by LPS and ATP. Remarkably, SR9009 also altered the expression of interleukin-6, known as IL-6, at the mRNA level, revealing the specificity of SR9009. While SR9009 exhibited noteworthy effects, it did not bring about a significant reduction of cytokine expression in RKO microglia, hinting at its specificity towards Rev-erb alpha. Expanding the anti-inflammatory potential of SR9009. Beyond just microglia, SR9009 also notably reduced the expressions of IL-6, TNF-alpha, and L1-beta stimulated by LPS in primary cultures rich in astrocytes. This suggests SR9009 has potential to act as an anti-inflammatory agent across various types of brain cells. Insights from in vivo experiments. The research progressed into an in vivo examination of the impact of SR9009 on LPS-stimulated neuroinflammation, particularly in the hippocampus of wild-type mice. These mice were pretreated with SR9009 before the injection of LPS. The result? The mice that had been pretreated with SR9009 exhibited a reduced response to LPS, as seen by the decreased levels of pro-inflammatory transcripts in the hippocampus. Synchronizing Rev-erb alpha and SR9009. The engrossing findings lead to the conclusion that while the elimination or knockout of Rev-erb alpha escalates inflammation within the hippocampus, it seems that activating Rev-erb alpha with SR909 appears to counteract this effect.